This is a time-lapse of live mesenchymal stem cells, imaged label-free using Nanolive's 3D Cell Explorer microscope. What you are looking at here is a refractive index map based on the cell's physical properties. In this presentation, we will cover some of the most popular applications for which Nanolive imaging provides a unique solution. Let's have a closer look at some cells. These pre-adipocyte cells were imaged for 48 hours using a compatible top-stage incubator, maintaining CO2, temperature, and humidity levels. Notice the cell at the top, how the nucleus spins and rotates right before division. This long time-lapse was possible since the system is gentle and label-free. Indeed, without fluorescent labels, you can bypass phototoxicity and photobleaching, which allows you to image unperturbed living cells for up to 1 week. Furthermore, due to the high lateral resolution of 200 nanometers and 3D reconstruction capability, quantitative data can be exported. The system can be coupled to a fluorescence module with up to 4 channels: DAPI, FITC, TRITC, and Sci5. This lets you localize proteins and confirm structures periodically, thus limiting the effects of phototoxicity. The fluorescent signals can be overlaid with the refractive index map for correlative imaging, as you can see here for mitochondria, lipid droplets, and lysosomes. Nanolive imaging is the only system that can image mitochondria without the use of markers or staining. Due to the high refractive index sensitivity of this microscope, the mitochondrial network can easily be distinguished at high resolution. You can see here the fine details of mitochondrial dynamics, including events of fission and fusion. The bright white dots that you see, those are lipid droplets. Due to their high optical density, these lipid droplets can be isolated and segmented in various cell types in 3D. Using Nanolive software for digital staining based on refractive index, you can export this information to your favorite open source software such as ImageJ to segment, track, and quantify the volume of these lipid droplets. For more information, you're welcome to read the application note concerning this use case, which you can find on Nanolive's website. It is also possible to apply perfusion systems to the incubation chamber, which can provide a great advantage when monitoring intracellular dynamics in living cells for drug discovery. Here, we have an example done by Chondrion, a pharmaceutical company focusing on mitochondrial diseases. Notice how Chondrion's drug candidate is seen rescuing the cytotoxic effect induced by buthionine sulfoxamine. Thanks to the automated stage, with 96 well-plate compatibility, the system is also suitable for high-content screening. The light source used for imaging is a gentle class 1 laser source, with a power of 20 milliwatts per centimeter squared, which corresponds to 1/5 of the solar power in Central Europe. This makes the system suitable for label-free imaging of cell types which are extremely sensitive to manipulation, such as stem cells and primary cells. In this video, you can see mitosis in mouse embryonic stem cells. The right panel in grayscale shows a Z projection of the refractive index map, and on the left, Nanolive software provides a 3D rendering based on the refractive index gradients. Notice how the cell adopts a spherical shape during mitosis. In the grayscale image, you can also distinguish chromatin condensation within the cell as it goes through metaphase and anaphase. After completing cytokinesis, you can appreciate fine filipodia formed by the cell membrane as the cell, now in interphase, adheres to the dish. The high temporal and spatial resolution of this system therefore enables you to observe interactions between cell types, which is especially important in the field of immuno-oncology. Here, the arrow points to T cells, detecting and killing a target cell. you can see how the intracellular components of the target cell slowly degrade, and typical apoptotic blebs form at the membrane. After the cell has died, the T cells detach, and this macrophage on the left clears up the cellular debris through phagocytosis. In this moment, you can appreciate the characteristic cell membrane ruffling of the macrophage. By using Nanolive's digital staining, you can get an immediate 3D volume reconstruction of cells without having to scan through the sample. Here is an example with T cells. From this quantitative information, you can then determine the dry mass, which then allows a characterization of different T cell populations. Please visit our website to read the application note concerning this. Thanks to the high imaging frequency and tomographic capability to visualize cells in 3D, Nanolive imaging can guarantee 3D monitoring of internalization processes, such as endocytosis and phagocytosis, which provides a global view of host-pathogen interactions. This is illustrated nicely with this example of human M1 macrophages phagocyting E. coli bacteria. On the right panel, the green arrow is pointing to a Listeria bacterium inside the host cell's cytoplasmic vacuole. We can observe this bacterium's rapid release through the plasma membrane and the host cell's death. As you have seen from this overview of applications, Nanolive imaging is the ultimate solution for studying unperturbed living cells. Indeed, it provides a harmless approach without phototoxicity or photobleaching, and without compromising temporal resolution. Furthermore, the design is simple, intuitive, and modular, so it can be adapted perfectly to individual research needs. With this brand new way of observing cells, you can now see what you've been missing. If you would like to know more about the many applications of Nanolive imaging, I invite you to contact us through our website, and you will be put in contact with your local Nanolive representative or distributor. Thank you for your attention.